begin today by making a number of clarifications. I've gotten so many questions on the Gram stain report and other things, so I thought I would try to clarify everything that you need to know to help you with that report. So firstly, in our formal description of the report, we do say that we want you to turn in a Gram stain slide with your report. However, due to our wonderful experience as of yet with our cell phones, we have decided to lessen that restriction and to say that if you turn in a really nice picture of your unknown Gram stain or your mixed Gram stain or whatever Gram stain you're writing the report on, we will allow you to turn in that photo instead of a slide. If you didn't get a good photo, then you do still need to turn in your slide. Please remember, and Doug was being super nice about offering to use his photo for other people to turn in their report because his photo is really rad. But please remember that your results are your results. So your descriptions need to be of your smear and your stain. And that's one of those cornerstones of science, right? Where we're saying that your ability to make good, detailed observations of the primary results, right, the results that you got in lab, is super important in that. So, by the way, I will be trying to make an Instagram site for our class so that when you're so stoked about your pictures, you can put them on there. And then I think we should have like a semester-long photo competition, and then at the end, whomever has the best photo by vote, I think, will get extra credit on their final exam. I think that will be a way to go, right? So I will get that going, and we're going to have like a rad photo competition. So maybe, Doug, you can win that one, and then you'll feel better about not sharing, right? Not sharing that picture. Okay, so does that clarify the restriction or the requirement on a slide? We cool? Oh, one more note on that. If you do turn in the cell phone picture, there are two things you must also do. One, your results must clearly state the observations that you made of that slide. Right? We want to hear about your description of what the slide looked like. So where the photo that you took might have been of one corner that looked really good, we would also like you to tell us that the middle was really jam-packed with bacteria, was way too thick, and you had to go way over to an edge to get a decent photograph. We would also like to know about that. So detailed observations of the whole darned thing, right? So that is a big part of your results sections, is writing those observations. And then lastly, in the discussion, could you also please include a discussion of what maybe could have been done better on the slide, techniques that you might use to improve, like, for example, not using the entire colony to make your smear, remembering that you might have a billion bacteria there, right? Yeah, please, Amanda.
I managed to destroy my slide when trying to get the oil off of it.
Yep.
So what should I do?
Slide completely decimated, destroyed.
There wasn't a whole
There's no
lot
hope.
to begin with.
Yeah.
So there's like nothing.
Okay, so, and you didn't photo it?
I did not.
And that's the one you know you want to report on?
Yes.
Do you have detailed observations and a detailed picture that you drew?
I did not do
Okay, well then probably you need to start that one over. Yeah. So if you don't have like any detailed observations of any kind, we're probably going to have to go back on that. Other questions? Other thoughts on that Gram stain report? Remember that assignment description is on page 29 and 30, so make sure you read through that, make sure you're comfortable with that. Oh, and also, I think Sam was just asking me about could she use MLA format for her citations? Absolutely. On this report, we are allowing you to choose your format, to choose your own format, and I don't care what format you use. Later in the semester, we will talk to you about standard science formats. And what's really kind of a bummer with science is that every darn journal uses a different format. So if you're submitting to the Journal of Bacteriology, you have to use the Journal of Bacteriology format. If you're submitting to the Journal of Infectious Disease, you have to use the Journal of Infectious Disease format. So it's not a standard, like unlike social sciences that use a standard format, science doesn't do that. So later in the semester, we will talk about that. But for now, whatever format you would like is fine. What are the questions on the Gram stain report? Doug?
So when we did our Gram stains, we didn't actually know what our unknown was.
Right.
And we figured that out this past
Right.
time. So are we still allowed to include like information about this previous lab, like how we discovered what it was in our Gram stain report, just to better enhance?
Sure, as long as that's like sort of there because of supplementary, that's not what the report's on, right? But certainly, yeah, certainly if it helps to enhance what you're writing, by all means, that is fantastic. Yeah, Julia. Yep. Right. So, okay, so what are we writing about the unknown from page 35, all that jazz, all those different results? That's her question. Oh, your specific stuff. Oh, yeah. So what about the unknown, though? That was your unknown, right? So that's not your Gram-Stain report. Gram-Stain report is just on one slide, whatever Gram-Stain you want to write it on, right? Just one slide. But what about the unknown? What are we doing for the unknown? That informal one, that informal press release or Harry Potter elixir potion or like wanted ad or whatever you want to do for your unknown. So if you had some weird mixed results for your unknown, what a better forum than in a creative sort of press release to talk about these crazy mixed results that you got. So there are no major restrictions there. So two different assignments going on here, right? We've got the Gram-Stain report that's a formal report on one Gram-Stain that you did. All the stuff about the unknown will go into that informal report that is sort of the press release. We feeling better? Okay, totes solid. Yes, Marissa. So your TA will decide whether they want you to upload it to Wild Courses or give you a hard copy. Probably they want a hard copy, though. I know a couple of them are in the back of the room. We can ask them. They decided to pop in. Okay, any other questions? Now, how about the exam? How are we feeling about exam prep? I'm not hearing much. It's Friday? Yeah. Okay, so exam prep is going to happen this weekend. Is that what I'm hearing? Okay, good. As you sit down to do your exam prep, go into the web page and under additional information and find the review. The review tells you what's covered on that. Today I will also tell you the end of coverage. Those of you who are loving the textbook representative chapters are Chapter 1, Appendix 1, and Chapter 3. However, please do know that the exam is over our lecture coverage. It is not specifically over the book. Now, the book does augment your knowledge. The other question that came up regarding that is that the exam is not specifically over lab concepts either. However, your knowledge from lab will augment your lecture knowledge, right, if that makes sense. So I'm not going to pull something out of your endospore lecture from yesterday in lab, but I might assume that you know that one bacterium we do work with is Bacillus subtilis, and I won't necessarily call on your specific knowledge, but I'll kind of know that you've worked with it, right? I'll assume you were kind of awake that day. And as long as you were awake that day, then I don't think you have any trouble, okay? All right. Good, good. Anything else? What else? Yes, Julia. It's not on Wild Courses at all. It's just on the open website under additional info. Yeah, yep. All right, are we good? Are we feeling good? Are we awake? Are we ready for prokaryotic cell flagella? Yes? Okay, we're going to talk flagella, prokaryotic cell flagella, that is. So we have now made our way through the internal nuances of, what did we name him? I can't remember. Norbert? Oh, Humphrey. Humphrey. We've now made our way through the internal nuances of Humphrey. But now we need to talk about one last external structure. Notice that Humphrey has a flagellum. This particular flagellum is called a polar flagellum, meaning that it's a single flagellum at the end of one side of the cell. So we also call this monotrichous. That is, there is a single flagellum. So I personally think you guys should make yourselves a Halloween costume with a monotrichous flagellum, and then you can mono-trick-or-treat. I thought of that right before class, though. That's what's real impressive. So in any case, bacterial flagella are whip-like structures, or excuse me, are propeller-like structures that are long hair-like projections. And you'll notice that they're split into three parts. When we look at this picture, we can see the flagellum inserted into the cell wall of what? A gram-positive or gram-negative bacterium? Gram-negative. How'd you know? Yeah, the cell wall's different. We've got an outer membrane. We've got a thin peptidoglycan layer. We've got bronzolipoprotein there. We've got lipopolysaccharide. We know that's a gram-negative cell, right? And we can see how that flagellum is inserted in it. Now, this flagellum has 3 parts to it. And forgive me, my pad just went out, so I'm going to label the parts on here. So these parts are the filament. So right here is the filament. This is the hook. And down here, this whole thing that inserts into the cell wall is called the basal body. So the hook, the filament, and the basal body. Notice, based on the hook, you can see why the movement of a bacterial flagellum is that of a propeller. So this flagellum can either go clockwise, or counterclockwise. And those are the motions available to it because it has got this hook that has sort of that curved structure. Now, the filament is made up of a protein called flagellin that rolls around to make a hollow cord structure. The basal body has a couple of different components. It has a ring called the L ring. It has a ring called the P ring. And then down at the very bottom, this whole kind of basal level set of rings is called the MS ring. They used to call them the S&M rings, but it was sad for me the day they stopped calling that because now I can't make tacky jokes as much about it stabilizing a whip-like structure. But in any case, what we can recognize is a different set of rings that are inserting down into that cell wall. So recognize then that this moves fast. These can spin really, really rapidly. In fact, we see Vibrio alginolyticus, which is a cool named bacterium that can move at 1,000. It can move its flagellum at 1,000 revolutions per second, propelling it along at lightning speeds. So, for example, I know, Sarah, you're over here. You like to run, right? And you're roughly 6 foot tall. Yes. So if Sarah could move as fast as Vibrio alginolyticus, she could run 82 miles per hour. And while I know she runs fast, she doesn't get up to those speeds very often, right? And so that is really, really quick. Now, we'll talk about in a minute how her movement differs from that of Vibrio alginolyticus. But we also can recognize first, before we mention more on that speed, that these can be virulence factors. So I have one of my precious giant stuffed microbes up here, and this one is Helicobacter pylori. Does anybody remember what that causes? It's implicated in, Stan? Peptic ulcers, though we do now understand that in the right balance, it helps to prevent acid reflux. So it's when it's off balance that it really starts causing peptic ulcers. But notice that it has a tuft of flagella at the top. This is actually something called Lophotrichus flagella. But what it does is it utilizes those fast-rotating propeller-like flagella to bore into your stomach lining. And that's what causes a lot of those symptoms of a peptic ulcer. So you guys can pass around this fella. He, at some point, when getting passed around, got himself a gangsta headband. But you can say hello. Maybe we should name him as well. Okay, so let's look at these different kinds of flagella. One at the end of a cell is monotrichous. If there's just a single one at one side, remember we called that polar. Yesterday, when you guys were in lab, you saw a bacterium with amphitrichous flagella. Some of you were a little bit confused about what you were seeing there because the cell itself was spiral, so the cell itself looked like that. And then out of both ends, you saw tufts of flagella. So I think a lot of your pictures in lab turned out looking like this, but you weren't sure if the whole thing was a flagellum or if what was coming out of the ends were flagella. And it's what's coming out of the ends that are flagella. So we might say that this particular spirilla actually had Lophotrichus, but also amphitrichous flagella. It had them out of both ends, both ends, but they were Lophotrichous. Peritrichous have flagella completely surrounding the cell. So we might talk about a very smooth criminal bacterium that we're going to work with later in the semester called Proteus mirabilis. And Proteus mirabilis is the quintessential causative agent of certain urinary tract infections. It has got a lot of flagella, and it gives it the capacity to move with what we call swarming motility. It literally means that if you put it onto a TSA plate in one spot, it will gradually swim in rings away from that spot, and it will completely cover the surface of the plate. So it is very swarmingly motile, so it's highly covered in flagella. So I know that Marissa had unknown number 140, right? Who else had unknown number 140, right? Okay, so all of you guys, you had a polar flagellum, one single flagellum in that bacterium. You didn't get to stain for it, right? But just so you know, that is one feature of your Pseudomonas, right? So get that jotted down. Peritrichus covering the entire surface of the cell. So we know that these flagella can move in two different possible ways. They can either move clockwise or they can move counterclockwise. So these two motions give rise to two separate actions. A counterclockwise motion causes them to move in a run. So when rotating counterclockwise, here we're showing obviously a peritrichus bacterium where you can see the flagella covering the entire surface of the cell. And if all of those are moving in a counterclockwise direction, you can see the bacterium moving along in a run. Here we see that they've moved to a clockwise rotation and they move around in a tumble. So a tumble is the way that bacteria change directions. So remember how I said that the way that Sarah moves when she runs, even if she's way slower than a bacterium, she has some advantages because she uses something called spatial sensing. That is, she's like, I'm going to run from here to Happy Jack. You know, I'm just going to take that Pilot Hill challenge, right? It's a good one. And she heads out on her run. She's using spatial sensing. That is, she knows I'm here and I want to go there, and I can see in space that that's where I want to head. However, bacteria don't have that leisure. They have to use what we call temporal sensing. That means they compare the environment that they're in now, and then they move, and they're like, oh, this is different. And they compare it to where they were before. And then they have to move again. So what do they have to constantly be doing if they're ever going to respond to a change in the environment? They have to constantly be moving, right? And if they stop moving, they don't know where to go. So it's the movement that allows them to get where they want to go. And that's that temporal sensing. Now, wait a minute, what are they responding to? What are they going towards? What are they going away from? Bridger, where's your, how does it know where to go? I mean, he's making love to the gangster headband. So, yeah, they go in response to predatory bacteria. Okay, what else? Elemental stresses, right? Okay, good. What else? Oh, come on, you guys. What do we move in response to half the time? Food. I mean, hello. So, in fact, bacteria are a lot like us. In fact, I was thinking this morning, they're way more like us than we would like to admit because they sense food at much lower concentrations than they sense repellents. So they will live in their own waste for much longer and not worry about it for much longer than they will live without food, right? They like to go towards food more than they like to go away from waste. But they respond to both. We could say that there are both chemoattractants and there are chemorepellents. So they are motile in response to these environmental factors, whether they be attractants or whether they be repellents. But attractants are sensed at concentrations as low as like 10 to the minus 8 molar. So they're very, very responsive to the food in their environment, whereas they're a little less responsive to the waste. And remember, they have to keep moving all the time to use that temporal sensing. So what we see down here is that there is a constant motion of a bacterium that is in the absence of any repellent or attractant. So it keeps on running, keeps on tumbling. It runs, it tumbles, it runs, it tumbles, it runs, it tumbles. And it keeps on doing that until it starts to sense that one environment that it was just in is different than the new environment that it now finds itself in. So if there is no attractant and there is no repellent, it moves completely randomly, but it keeps on moving. So we know that the chemical attractant or repellent must be sensed by the cell in some way. And the way that it's sensed is through chemoreceptors. So there are these receptor, as you guys talked about earlier, was it this week? That certain transmembrane proteins are going to be receptors, right, that trigger changes within the cell. So these receptors will trigger that motion. And it's actually really interesting to note that there are proteins that we're not showing here next to flagella that then change the direction of rotation of the flagellum, but they also play a role in allowing the proton motive force to come swishing in, and that's what drives that propeller-like rotation. So chemoreceptors are really important in allowing that movement to take place. So as I said, there's these equal numbers of runs and tumbles. Bacterium moves randomly when there is no chemical attractant or repellent. But say all of a sudden there is food in the environment. Maybe there's a glucose gradient. One of scientists' favorite experiments is to put some sort of chemoattractant in a capillary tube and then put that capillary tube in one spot on a plate and then see how bacteria move towards that chemoattractant and be able to monitor its motion. So say there suddenly is this glucose chemoattractant. Now the bacteria sense that one environment where they were before is not the same as the new environment that they are, and so they stop tumbling as frequently. So what does that have the effect of doing if they stop tumbling as frequently? They go. Good. Yeah. Oh, brilliant. You got it. They go. And that is to say, another way, the runs get longer relatively. Yeah, exactly. And so we start to see something that is termed a biased random walk. The bacteria get there. They just don't get there in a very straight path. It's a darn good thing they're so fast, right? Because they take a path that looks sort of like this, where there's still runs and tumbles, but when the tumbles become less frequent, we start to see the movement in the direction of the chemoattractant or away from the chemorepellent. And this, as I said, is called a biased random walk. So who has questions in the way that flagella coordinate the motion, I guess, of a motile bacterium? Of course, remember that not all bacteria are motile, right? Not all of them actually have flagella. Only some of them do. Good? Solid? Yeah, they can, Megan. It's a great question. But more often, gram-negatives are motile. And I think in a lot of ways that, well, and so when I say that, it's like we say that with the caveat that we really don't know about a big majority of the bacterial population, right? You know, we've culture characterized less than 1%. But with regards to those that tend to be the famous bugs, you can think about why many of the gram-negatives that live in the enteric region may be causative agents of UTIs, why they've adapted to have a lot of motility, whereas more of our gram-positives that live, you know, nasal pharyngeal, they don't need as much with regards to motility. Yeah. Good question. Very good question. You could likewise put in that blank, moves away from the repellent. So we are nearing the end of the exam coverage. We actually only have a couple more little things to talk about, and then I'll let you know that we've hit the end of what you would see on your Tuesday exam. So this is a picture where you can see this bacterium has flagella. These are the flagella right here. So these really long structures, those are the filament of the flagellum, and of course comprised of that protein flagellin. But notice that also on this bacterial cell, there are these little guys. They look like little hairs coming off of the cell. And those are called fimbriae. They're different than flagella. Fimbriae are they're much smaller. They have an internal hollow structure as well that's helical, but they're not made up of the same protein. So despite having similar ultrastructures, they are definitely shorter and thinner. But what they do is really interesting. Fimbriae often allow for cells to adhere to certain surfaces. And depending upon the resource that you're reading, sometimes fimbriae are also known simply as pili. Your book differentiates them. It calls everything fimbriae unless they're involved in conjugation, and then it calls them sex pili. Okay? Right? You guys remember that process of swapping genes, right? Sending plasmids across the sex pilus. Well, your book says that they reserve that term, pili, for just sex pili. And then they use fimbriae for everything else. So what are some other functions of fimbriae? Well, these fimbriae can attach to surfaces. So on the surface of a little fimbriae, you might see an adhesin molecule that would attach to a surface glycoprotein, say, for example, in your urinary tract, and then it would allow the bacteria to hold on to that surface even when the urine comes rushing through. So we could say that fimbriae, like flagella, can be what? If they make the bacteria more pathogenic. Virulence factors. Nice, Jojo. You're on today. You're just like bing, bing. She's been studying already. So these are going to allow them to adhere to specific surfaces. The other thing that they do is a weird kind of motion, actually. They can allow this short kind of twitching motion that you see in certain bacterial genera. For example, myxobacteria, they can move in these short twitching motions across a solid surface. And when they move like that, we call them Wolfpacks. Literally, that is the technical term for it, Wolfpacks. So they move along in these like short twitching motions, kind of quivering movements across a solid media surface. Lastly, as I mentioned, the term sex pili in your book is reserved for the, if you will, the type of structure that forms that is very much like this, but is dedicated to the transfer of DNA from a donor cell to a recipient cell. For example, the transfer of an R plasmid from one bacterial cell to another. This ends our coverage for the exam. Who's got questions? Anything just pop into your mind while you were in that portion of the lecture? Okay. Yeah, please.
Is there ever a problem with a cell having too many flagella?
Too many flagella, I reckon. You're a guest today, right? Yeah, it's nice to meet you. Hi. Too many flagella. No. I mean, well, I guess you're asking who has the problem, right? I guess we could say we have a problem if we have a bacterium that is swarmingly motile and it can really get around and cause like a systemic infection. So yeah, then maybe you could consider that a problem for us. I wonder, oh, well, what if we had overexpression of flagella? Does anybody, I don't know, want to comment on that? Could it ever cause the bacterium problems? I don't know. Maybe. If you just had so many. I mean, what about overexpression of sex pili, right? I mean, that's the thing. No, I really, I don't have a good answer for that, but it's a great question. So, anybody else? Okay. Yeah. Rachel.
When you transfer a plasmid or whatever, does it replicate it by standard DNA?
Yeah, oh God. So she's asking a super, super good question that I promise we'll talk about the details later. Rachel's question is, when one bacterium, the donor bacterium, makes a sex pilus and connects itself to a recipient bacterium, and it transfers an R plasmid across from the donor to the recipient. Now, remember that the donor bacterium keeps the plasmid too. It only transfers a copy to the recipient. That's why this can be so problematic with an R plasmid, right? So both of them after the transfer will be resistant. And the question she asked is, does it use a standard type of replication to enable the copy to go to the recipient cell? And the answer is no. It uses something called a rolling circle mechanism of replication. And I promise you later I will cover that better. So it's a fantastic question. And this means that we get to dive in and spend the rest of our Friday talking about something that's not going to be on the exam. And that is that we get to look at a eukaryotic cell. So I also have a model of our eukaryotic cell. You can be pondering good names for it as well. But I want to talk about some of the differences, if you will, between these types of cells. But I want to center our discussion on the microbial members of the domain eukarya, because all your other classes talk about cells like our human cells that are eukaryotic. So we'll probably be focusing a lot more on that. But to begin us off, I've got some pictures of the diversity of eukaryotic cells. They can range all the way from, say, our cheek cell, this is a picture of a cheek cell, to down here, these are called diatoms. Diatoms are a type of algae. Algae is not a technical term anymore, but we can say that they are photosynthetic protists. That is, they produce chlorophyll. They're able to synthesize organic carbon. You can see the pigmentation in them, but what you're also noticing about them is they have a wicked cool cell wall, and that cell wall is composed of peptidoglycan, right? No. Oh, almost gotcha. Yeah, no, not so much, right? Because we know peptidoglycan is only found where? Bacteria, you got it. So it actually is composed of silicon dioxide, which maybe our chemical engineers will tell us looks like glass. It's actually looks like glass. And actually this was really cool because diatoms can, have you guys heard of diatomaceous earth? Diatomaceous earth is actually a common product in many abrasives or paint removers or even insecticides, because the cell wall breaks down into these extremely sharp pieces, right? It's like glass, and it can be used as an abrasive, or it's even in some toothpaste, I think. So it can be used like that and to kill insects because of the sharp edges. So they have silicon dioxide cell walls. Now that contrasts with a yeast cell over here, and this yeast cell, it has a cell wall too, but it's made up of cellulose or chitin, no peptidoglycan. Up here is, actually this is a ciliophora. It's called paramecium. You guys may have heard of it before. It's covered in cilia, so no pili, right? No fimbriae. But in eukaryotes we see cilia. It's actually got no cell wall, and in fact it's flexible to have the ability to have a cytosome or cell mouth that it can invaginate the food that it needs and be very malleable and flexible in that way. So point of me showing you this slide was not to yammer on about these really cool organisms, which is cool, but is to tell you that there's a lot of diversity in the eukaryotic world. So when we make generalizations about the cell structure, remember that there will be exceptions to every rule. But that being said, let's jump in. Eukaryotic microorganisms can be split into two groups, the fungi and the protists. So I want to make sure everybody's with me on this, in that we're getting the so-called taxonomy of life here, and recognizing that we have now moved from one domain of life to another. So recalling that our three domains of life were what? Bacteria, which we've centered much of our coverage on, and archaea. Now, we're going to talk more about archaea later. And finally now, we're switching to eukarya. So as we look at this picture, we can now say that this domain of life can be split into two microbial groups, the fungi and the protists. Now, you guys have already worked with a type of fungus in lab. What is that, Julia? You bet, which accidentally showed up on her unknown slides multiple times. So she can tell us that the type of fungi that we have worked with are yeast. Okay, we will later look at some other kinds. So this is our grouping. Now, protists classically have been split into the algae and the protozoa. We could broadly say that the algae are photosynthetic protists and the protozoa eat other stuff. That is, algae are self-feeders, protozoa are eaters. But now we don't really have a legitimate taxonomic difference between the two, and that's why I said these terms are no longer considered technical terms, because really some algae are more closely related to protozoa phylogenetically, that is by virtue of genes, than they are to other algae. So it's confusing in that although we tend to talk about those two groupings, when we think about their genes and their relationship evolutionarily, it's not really a legitimate difference, okay? So let's talk just a little bit more about the structure of a eukaryotic cell. And you all will tell me that Humphrey Herbert? See, I can remember your names, but this guy just doesn't have any personality. It's sort of rigid. So this guy had structure and rigidity, right, based upon what? Cell wall, right? But what about, say, our paramecium? Where does the rigidity and structure of the cell come from? Cytoskeleton. Good. Is that what you're going to say, Christian? Good. So the cytoskeleton. So we actually start looking more internally for the structure of a eukaryote. Now that's a sort of broad statement because we know that yeast have a cell wall and diatoms have a cell wall. But we also tend to associate eukaryotes with a cytoskeleton. And that cytoskeleton has three different components. The first component is called the microtubule. And the microtubule is comprised of a protein called tubulin. And the protein tubulin has two subunits, or said another way, it is comprised of two polypeptide chains that interact with one another. Said another way, tubulin has what level of structure? Yeah, good. Who was that? Yeah, very nice. Rex is on today as well. So Jojo and Rex got enough sleep last night. You guys weren't down at Crowbar, were you? So this is the largest structural element. It has quaternary structure. It is comprised of both alpha and beta tubulin. They wrap around in sort of a helical way to make a hollow cored protein. Really has a lot of strength, but it can serve multiple purposes within the cell. So, for example, they form mitotic spindles and cilia and flagella. They help to form pseudopodia. They are really important in something called axopodia, which if you've ever seen a beautiful marine protist that looked like a star, those are axopodia, and those axopodia allow marine protists to have buoyancy in marine water, which is really a neat adaptation. All of those have these microtubules to thank for that. Microtubules further form what I like to think of as the highways of eukaryotic cells. In fact, I find it very tempting to try to make an analogy of a eukaryotic cell to a city that never sleeps. And in this city, the highways are the microtubules, and the cars or the taxi cabs that are on the highways are vesicles. So these vesicles carrying components are streaking down the highways, the microtubules, and they're delivering things to various places, maybe from one organelle to another organelle. So these provide the highways along which those vesicles can travel. It's very interesting that in certain cellular systems, they've done some interesting studies where if they take away the ability of a cell to make these microtubules, then the cell actually not only, well, obviously it loses shape, right? It loses its shape because of the loss of the microtubules. But those of you who remember that foundational rule of biology, and really it's one of chemistry as well, will recall that if they lose their shape, what also happens? Yeah, they lose their function. So if form is what dictates function, then the loss of microtubules, the loss of form, means the loss of function. This has been shown many times in neural cells, actually, which is interesting, right? Cool. So what else is part of a cytoskeleton? Microtubules is? What else? Microfilaments. Yeah, dang girl. Microfilaments, you bet. So now these are smaller. Did you notice that the microtubules were 25 nanometers across in diameter, but a microfilament is only 4 to 6. So it's very, very much smaller. 4 to 7, I guess. But it's very important in motion, and for example, it's very important in the formation of pseudopodia. So remember that pseudopodia are these long surface projections of a cell. We talked about them a tiny bit with respect to white blood cells that phagocytose bacteria. We were actually mentioning that white blood cells try to phagocytose bacteria with capsules, but can't get a grip on them. So that process of phagocytosis is enacted by pseudopodia, right, forming. And this particular image is actually a video that I'm going to play. And what it's showing is an amoeba. So like white blood cells, amoeba are also capable of forming these pseudopodia to try to reach out and bring in their food. So you can see actually right here that this particular amoeba is trying to reach out and grab in some nutrients via phagocytosis. And so we'll watch this quick video clip, but what I want you guys to realize is that as these pseudopods stretch out and get longer, they actually are, it's actually actin polymerizing in there. And then when they reach back in, it's depolymerizing. So actin polymerization is what's leading to that, that motion. So we'll get that going. Sorry, there we go. Oh yeah, let's do it again. See, backwards. Depolymerization. Polymerization. Yeah, right. Well, oh, one more time, just for Esteban. Okay. Right? I mean, it's addictive. We could just do this all day. It must be a Friday. So this is pretty cool. But another thing that you're going to find even cooler, maybe, is that there are certain pathogens. Have you guys heard of Listeria monocytogenes? Yeah. So Andre is shaking his head yes. Do you remember anything about it? Not at all. No? Let me give you a hint. Cantaloupe. Anybody remember that? Yeah, the Rocky Ford cantaloupes, which it was kind of sad because Rocky Ford cantaloupes were a part of my childhood. You know, we all grew up eating Rocky Ford cantaloupes. And what happened was that Listeria, which is a common pathogen of raw hot dogs and like raw deli meat and things like that, Listeria is often found on those. I used to love to eat raw hot dogs. That was my Saturday morning treat. I would get up at 5 and watch cartoons and eat raw hot dogs. But that tells you something about me. I didn't get Listeria. I did not get Listeria, but a lot of people did get Listeria when they ate those Rocky Ford cantaloupes in the fall of 2011, right? And what happens is that Listeria invades the body, and once it is inside, it triggers your cells to polymerize their actin, and it jumps a ride on it. Not kidding. It actually hops a ride on polymerizing actin, and that enables it to get from one spot to another. Yeah, isn't that cool? It sends out a signal that says, polymerize your actin, I'm going to hop a ride. And it does. And that is part of its pathogenesis. So it's really an interesting pathogen in that way. So lastly, let's end our coverage of the cytoskeleton by looking at intermediate filaments. These are comprised of protein part of it is made of keratin. They are very stable and very supportive. They tend to look like ropes. I don't have a picture of it here. They're less well characterized than the others, but you can notice that they are intermediate in size, right? Relative to a microfilament and a microtubule, they're approximately 10 nanometers, which means that they're kind of halfway in between with respect to size there. They're really structural. So one of the places that we know we see them is in eukaryotic cells. We see them just subsurface of the nuclear membrane, where they provide a lot of structure to that nuclear membrane. So if you're envisioning sort of some of the structure of the nucleus of a eukaryote, some of that comes from microtubules. So that is one area that we know they're playing a role. Okay? Any questions on the cytoskeleton of a eukaryotic cell? Solid? Okay. Yes, literally and figuratively. So notice then that in the nucleus, we've got many layers and many pertinent structures, but the first one is the nuclear envelope. So the outer nuclear membrane and the inner nuclear membrane together comprise the nuclear envelope. So you might think that a nucleus, did we come up with a name for it yet? Babe? Dave. Dave. Do I hear Dave? Oh, Sully. Okay. Sully Dave. No. All right, we'll call it Sully. So in any case, you might think that this nucleus would be really impermeable to a lot of substances because it's got both an inner and an outer membrane. But notice that there are areas that are spanned by something called a nuclear pore complex. And the nuclear pore complex lets just about anything in. So it turns out that the nucleus is kind of a party house. Everybody come on in, right? Proteins, ribosomal subunits, free passage for a lot of things. The nuclear pore complex, while it is a little selective, is not very selective. Nowhere near the kind of selectivity that we see for the cellular membrane. So this nuclear pore complex spans and makes a route of transport through from the nucleus and out into the cytosol. And as I mentioned, just subsurface of that nuclear envelope, we get a lot of intermediate filaments forming the nuclear lamina. This has been clearly observed in animal cells, but it could be that even right now, if you happen to be dorking out on your computer, Justin can check for us and see if they have observed a nuclear lamina in certain microbial life, maybe in your algae or protozoa or what have you. Ashley?
Is that kind of similar to the cell envelope in India?
Yeah, I mean, so it is, right? We've talked a lot about within bacterial cells having multiple layers of membranes, for example, in your gram-negative as well. So, yeah, which starts us thinking, doesn't it? And maybe you can continue pondering that concept of similarities that we might see within internal structures within eukaryotes, similarities to bacterial cells, and keep mulling over those comparisons. I like it a lot. Now, notice that inside of the nucleus, there is this kind of discrete region. It's not a membrane-bound region, but depending upon what the cell's doing at the time, it's very observable. So this is something called the nucleolus. It's really distinct, but it's not membrane-bound. So what happens here is something super important. It's where our RNAs are made and the site where ribosomes are partially assembled. So let's get this down, because this is super important. A ribosome is what? Protein maker. Good. I like it. A protein maker. And what is a ribosome composed of? RNA and protein. Good. Or said another way, in a very broad way, we could say it's comprised of nucleic acid and protein. So it is comprised, then each of the components of the ribosome, the RNA, what is that made up of? What is RNA made up of? Nucleotides, bound together by what kind of bonds? Oh dear, not so much peptide, but Phosphodiester. Oh, but on the other hand, the proteins that are in it are comprised of amino acids linked together by peptide bonds. Okay, now we're going to rage that question on the exam or what? Okay, yep, all right, I thought so. Okay, so in any case, this is the site of rRNA synthesis in a eukaryote. And it's actually maybe a good time to mention that ribosomes in a eukaryote are different than ribosomes in a prokaryote. They're 80S rather than 70S. But this is the site of that rRNA synthesis and partial assembly of ribosomes. And then those ribosomes can head on out and do their work in protein synthesis out in the cytosol. So they don't synthesize proteins in the nucleus, they synthesize proteins out in the cytosol. Now, further, the kind of stuff that looks like, wow, Rachel just got a lot of static in the inside of the nucleus, that's actually chromatin. Chromatin is the genetic material. Oh, I wish she canceled, you guys. I am so devastated. I was supposed to have a massage today. She canceled. Anyway, you don't need to know about that trauma. Euchromatin, yeah. Euchromatin is the highly relaxed chromatin. That is, it is the chromatin that is being wildly expressed. The DNA, remember the central dogma, the DNA is getting converted to RNA, is getting converted to protein. In euchromatin, that's happening wildly because it's open and accessible. In heterochromatin, it's very tightly packaged, right, all wound up, and that's not very active, right, actively expressed. So one of the things that came up some time ago is something called the accessibility hypothesis, and the idea that in heterochromatin, these genes don't get read as often. And now we're actually starting to talk about something a lot called epigenetics that's somewhat related. I want you guys to I want you to ponder that really cool concept. So, you know, tonight at the bar, when things really get boring, pull it up on your cell phone. Don't you love doing that? And then you look like really important. You're texting, you're like, so, you know, epigenetics. Okay, good. Have a great day, you guys. It's on the regular web page, Jim.